Okay, let's start. In the case someone joins us, we'll be here to answer the questions. So, my name is Burhan. I'm from Kazakhstan. Recently, I did my master's at the University of Edinburgh. Currently, I'm doing my PhD at the Nazarbayev University and working as a teaching assistant. Also, my research interest is regarding the viral helicases as a therapeutic target, and currently I'm working on SARS CoV-2 and SP13. It's enzyme against which we would like to find a drug. So today we'll be discussing drug development process against viruses and how it can be different from the drugs against bacteria or other species. Okay, so today we are going to briefly talk about viruses, compare them with bacteria, define how it could infect our body and the viral structure, and understand how we can block their activity in our body. So, first question would be, what is the difference between bacteria and viruses? So, in a table form, if we discuss, so we know that viruses, here viruses, this is one type of virus, it's influenza type virus, and here the cell of bacteria, one type of bacteria. So if we think viruses are non-cellular organisms, because here you can see that the outer part, there are spike proteins, the shell, this is genetic material, but it's not cell structure, and this is a cellular structure, but also it differs from the animal and plant cell. So nucleus, they both do not have nucleus. As you know, ribosomes, bacteria do have their own ribosome structure. They both have genetic material. Bacteria can reproduce themselves. There are many ways bacteria can reproduce, and viruses, they can replicate only inside the host cell. So host cell could be bacteria, could be animal, could be plant cell. Regarding the treatment, so it depends on the type, but mainly the treatment is to kill them or to prevent their activity, to prevent their reproduction in the host body. Benefits, so bacteria, there are some useful, helpful bacteria in human body that help to digest some food. Regarding the viruses, you could use some viruses that kill bacteria as a therapeutic, but apart from that, viruses do not have benefit. Only it's beneficial when you use for a purpose. Apart from that, viruses could be different depending on their host organism. Size of bacteria is also smaller than animal cell, smaller than plant cell. Viral structure is, I don't know, many times smaller comparing to the bacteria. Is it alive? So bacteria is alive, and viruses can be activated only inside the host cell. You can learn and extend your knowledge regarding the difference between them by just clicking to this link. Okay, so I would say let's discuss what is a virus and are they living creatures, but I have already answered for this question. So do you know some common vaccination to certain disease caused by viruses? So we'll come for this question, how we can prevent their activity and how we develop a novel drug for certain kind of pathogens. And I don't think that there's a difference between producing a drug for any type of organisms. It's just in molecular level they are pretty much the same studies, but then when you try to implement this drug, there can be some differences. Testing drug to bacterium is easier, as you can visually see them, but with viruses it's different, and it would be more difficult to test. Okay, viruses, historically it was found in 19th century, around 1990s. So firstly they found a plant virus, then investigating they found other type of viruses. So the most, because nowadays we're experiencing this COVID pandemic, the first pandemic that regarding viruses we have seen. It was in 19th century, in the first quartile of the 9th century, the flu. It was firstly written as Spanish flu. So millions of people died from this flu because it was infectious, and it spread to the other parts of the world, also not only in Europe. So inside the so from this structure you can see these are the spike proteins. These proteins come to the cell and they have interaction with our proteins in our cell membrane, and so for example COVID-19 SARS CoV-2 spikes have pass cell membranes through ACE signaling pathway. They have this interaction with this protein, and then they can release the genetic material inside the cell. So genetic material can be easily replicated. So difference of SARS CoV-2 from other type of viruses, so they can be positive and negative. So those are positive, it means that they can be translated and then easily translated, but with negative form, for example flu, genetic material is negative, so there should be converted to DNA, then it just can be replicated. So this is the differences. Okay, the genetic material could encode different proteins. So today I'm going to talk about non-structural protein 13, which is enzyme, helicase enzyme. Helicase enzyme is unbinding double-stranded genetic material. What it is needed for, if it is not unwinded, it cannot be copied, because if you do remember the replication process of DNA or double-stranded RNA, it should be unwinded, and only one strand of this genetic material could be replicated by DNA or RNA replicase. So that's why unwinding process is very important, and helicase here have a big role. And how we can stop these viruses spreading. So scientists and drug developers mostly target different part of these viruses, so the drug could just stop spreading of the virus or stop the viral activity. So let's take firstly, mostly they try to find a drug against these spike proteins, so they wouldn't interact with receptors on the surface of cell membrane. But the problem with these spike proteins, they are mutating, and that's why the drug that they found could be in the future not specific. So and that's why we try to develop a drug for different kind of proteins. For example, I'm going to talk about the Helicase enzyme, which have less mutation comparing to the spike proteins, and its good part it's have conserved regions. Now I'm going to show you conserved regions of helicases. So helicases, as I said, they're enzymes which are responsible for unwinding the nucleic acid structures. This is our DNAs and RNAs, and they are very essential proteins. If their activity could be blocked, it means the spread of the viruses could be blocked also. Okay, let's look for the protein structure. Sorry, my computer runs too slow. I'll come this. So as I said that they have conserved regions. So what is this? This is a protein sequence. It means the sequence of amino acids in the helicase enzyme. Here are the most known 5 human coronaviruses. So SARS CoV-2 is currently causing the pandemic all over the world. SARS CoV-1 caused epidemic in 2002. MERS-CoV caused it around 2010 in South Korea. And there is Hong Kong coronavirus and HU coronavirus. And you can see these black regions. Black regions, it means these parts are the same. So the same, meaning that they have these conserved regions. So if we will find, let's say, a molecule, a drug that would bind this part, it means it's going to be specific for all these coronaviruses. And this is the, let's say, code or amino acid sequence of the helicases, and you can see that there are many parts are pretty much identical. So how we study them? This is the protein structure. As you can see, it has different domains, and these we will choose a binding site of this domain to which we can find a drug where and dock them. So if a molecule drug could interact, could bind to this part, it could inhibit its activity. So it means that how these enzymes work. Actually, this enzyme, helicase, firstly binds to the ATP or other GTP, it can be other NTPs. Then it activates itself with phosphorylation, and then it starts unbinding because it needs energy to unwind these nucleic acids. So if we stop its binding to this ATP or GTP, it could not go further and do its job. So this is our actually job. So, but the proteins could have different binding sites. So it is important to find a binding site which would be more specifically good binding. So here, for example, in ATP binding sites, here it is identified that, for example, glutamate 375 and, for example, arginine 567, lysine 288. After identifying this pocket, we just using a computer programs try to identify best suitable molecule that could bind here, and it would stop the activity of this protein. So after we find many suitable drugs, here we test them. After testing them, we just can use them at the bench in laboratory. We can buy these molecules, express the protein itself, and test if our findings on computer-based programs are identical with the experiments that we are going to do on the lab. If it says that it's okay, so we could assume that our findings and we can suggest that it is going to be good drug against the pathogen that we were studying. So which programs we can use here? So firstly, for studying the sequences, we can just download, it's freely available, protein sequences. Then we have programs like Mimi Suite, and then we can use Sprite to find suitable drugs. And studying the protein structure with PyMol, it's another program. We can find a suitable binding site, study it, and with docking programs like Auto.Vina, we can see how it is suitable. At the end, we just study the molecule, get it, and do experiments on lab. So all the computer programs that I mentioned, most of them are freely available, and if you contact me, we can further study together, and because it's not very difficult to do this drug development yourself on computer-based programs. So this is the schematic representation of how further studies could be done. So you will obtain this molecule, and then you will study helicase activity. So the genetic material, you would use the fluorescence dye, and it will give you a green color or blue color. Then if you use helicase with ATP, it should unwind, and the genetic material would not give you color anymore. Then you would use the drug with a certain amount, and you would test if the helicase activity is still there. Is helicase is still active? If it's not active, or if it's less active, we could assume that our drugs is working well. So here the representation of how it could be tested. So bound DNA, free DNA. Here when you are increasing the amount of helicase, you can see that there's becoming less free DNA and more bound DNA. Here you can see the other experiment where there is double-stranded RNA and single-stranded RNA, and when you increase the amount of helicase, we can see that the amount of the single-stranded RNA is becoming more, it means the helicase is active. Same conditions, you can test the drug, put the drugs here, and in this amount of helicase, if it is stopping the helicase activity, we can assume that the drug is working. So how many years we need to find a novel drug? So the part that I'm talking about is around 3 years or less, because this is mostly computer based and on the laboratory with simple experiments on bacteria and etc. It can be on 3 to 5 years with a good drug, with a clear molecule, and then it has this regulatory experiments, preclinical, clinical, and tests which are done to human. So it should be safe and it should have no other side effects to human, and there will be other regulatory approval, and total we could say normally it takes 12 years to develop a drug. But when they were forced to have a good drug in a small time period, for example, Ebola had vaccination in 5 years, and we can see there that they supported with the regulatory part, also in preclinical and clinical trial parts, they were also supported. That's why they could do that in small time period. The drug discovery part, actually, it could be decreased till to 1 year, or if they have some drugs prepared before that could be used for novel things, it could be less than like 6 months or so. But the preclinical, clinical parts are very important to test for safety and for side effects, for toxicity and other things, and if it is specifically working on people, human body, and etc. We were assuming that there will be people from Albion, but because they are not here present, I would send an email, and you can just send your questions, and I can answer them. Thank you for your attention.